Clonal selection theory explains how the immune system responds to an infection and how certain type of B and T cells are selected for destruction of antigens. The first process is called maturation. B cells, also known as B lymphocytes, are a type of white blood cell of the lymphocyte subtype mature in red bone marrow, which is at the core of most bones. Acquire B cell receptor surface antibody to a specific antigen. T cell, also called T lymphocyte, which matures in thymus gland. Lymphocytes that has differential in the thymus gland where it requires a T cell receptor. Daughter cell that grows up in the bone marrow is different from its parents and is also different from all of its siblings. This application applies for both B cell and T cell. B cell and T cells have receptors that later on will be their antibodies. Daughter B and T cells will have a slight change in DNA which result in different receptor for each cell to identify their enemies in a human body. To be clear, this B cell does not have the same receptor as than this cell. This B cell's receptor is different from this B cell's receptor. Therefore, all of them have unique receptors, including with the parent. All the receptors have different and unique pattern. This application also applies for T cell. This T cell have different receptor compared to this T cell and compared to the others, including the parent cell. Next, this step is for the binding of the receptor to the specific antigen. To be in general, to find the specific receptor to attach with the specific antigen to destroy that bacterium. Once B and T cells are ready, they will migrate to the lymph nodes all over the body. Let's take an example, armpit. Those cells will migrate to the lymph node and wait there. They come here into this lymphatic vessel and wait for the infection to occur. When there is an infection, for example, these cells have been infected by certain bacteria. For example, in those cells which are a part of the immune system, which are called dendritic cells, and these cells can eat some of those bacteria and ingest them. But the main point is actually to present this bacteria to the B and T cells. They come here. When dendritic cell ingests some of the bacterium, they come here, which start here in tissue, and they go up to the lymph nodes, where it transfers the bacterium, or sometimes others escape to the lymph nodes and enter here. Where they are going to present this bacteria to these cells. And we also have dendritic cells here, which means that they have this protein called MHC on which they present piece of this bacteria that they have ingested. Now, T cells here are unable to identify this bacteria when they are just freely floating. They need this antigen presenting cell to show this piece of bacteria to them. Only T cells can recognize this very specific pathogen and are going to see it and bind it and react to it. Meanwhile, these B cells are made to react directly to this bacteria without this antigen presenting cell. And so the specific one that is specific to the bacterium is going to see it, bind it, and also be alerted. Therefore, the other B cells will not be used anymore because their receptors are not suitable for this receptor of this bacterium. So, this B cell and T cell will be neglected, and this B cell and T cell will be replicated more. We are going to focus on activation of B cells and T cells to replicate more with the same receptor. Now, B and T cells are going to be activated and starts replicating, and each of the daughter cells going to have the same receptor. This B cell had the same receptor as this B cell, and there goes T cell also had the same receptor with this T cell, and compared to all. Some of them will become effector to fight infections, and other will become memory cells, which they are going to wait until the next time that this pathogen comes around. Okay, we are back to the lymph node. The B cells are going to pump out antibodies to attack this bacterium, and the T cells, some of them are going to stay in the lymph nodes, and some of them are going to travel out to this side to join the battle, because here the infection is still going on. So this T cell will have to fight this infection, while this B cell, which has the same receptor, will invade those bacteria. So this clonal section, and the reason it's called clonal section is because you're going to select B and T cell that you need, and then you're going to start cloning them like crazy into this army. And the process involves all these very unique specific B and T cells coming from the bone marrow and thymus to the lymph node, where they are going to be met by these bacteria that are infecting your skin, that are going to travel through the lymphatic vessels to the lymph nodes. And so they will meet there, and then the armies will be prepared. Thank you.